Hello. Today's lesson is on the role of neurotransmitters in weight regulation. There are more than 50 neurotransmitters and their receptors near the hypothalamus that serve as the brain's messengers regulating food consumption. Orexigenic neurotransmitters are combinations of peptides and hormones that stimulate feelings of hunger and appetite. Anorexigenic neurotransmitters are neuropeptides and catecholamines that curtail the desire to eat and promote weight loss. These have predominantly been studied in animals, particularly mice, with studies increasing in humans. Some potential targets for weight intervention include norepinephrine. It triggers cravings for carbs and is not satisfied by small portions of food. It demands more and leads to diet-induced thermogenesis. Insulin, when blood glucose is low, decreases the desire for pleasurable fatty foods, and problems with insulin production contribute to weight gain. MCH causes one to eat. Mice who overproduce it overeat, and those that lack MCH are thin. Overexpression of MCH also reduces glucose tolerance, provokes insulin resistance, and causes depression. Dynorphin is implicated in stress-induced eating. Some of these neurotransmitters work in opposition of one another. For example, some neuropeptides derived from POMC contradict the desire to eat triggered by MCH. The protein leptin triggers feelings of satiety, while ghrelin signals hunger. Potential solutions related to neurotransmitters in addressing excessive weight include changing the mindset regarding weight. Think more about losing fat than dropping pounds. As we age, diets to shed pounds will not work in the long run, as we cannot shed fat cells, but we can reduce their size. As several orexigenic and anorexigenic neurotransmitters function to turn on and off food cues, scientists can explore whether altering any or using them to balance each other can improve weight control. There has been promising research on MCHR antagonists working positively against obesity, anxiety, and depression. The MCHR1 gene, a receptor of MCH, might be one target to curb hunger, appetite, and increasing body weight. Leptin, as a satiety component, is also a possibility. When injected in obese mice, leptin reduced 40% of their weight, mostly in fat. This is the opposite of ghrelin. While norepinephrine demands large quantities of food, serotonin is an appetite suppressant. Another form of balance can be created via non-homeostatic mechanisms. Scientists are studying the prefrontal cortex, for which connections with the hypothalamus allow them to override hunger. The circumventricular organs in the brain and the limbic system are also avenues to explore more deeply in controlling obesity. Thank you for tuning in to this Health 570 presentation for strategies in weight control. Feel free to send me any questions. Thank you.